right, we're going to get started in just a minute. We can get settled. Thank you all for coming out. My name is Timothy Cardibona. I'm the Vice President of Public Policy for Connect. We are originally based out of San Diego. We just opened our Washington, D.C. office about a year ago. And our main mission is to educate Washington, both Capitol Hill and the administration on innovation. Innovation is a big buzzword right now, and we think it should be the big buzzword, but we think there's still a lot to learn about how it works. And so today, part of our mission is to bring startup companies to the Hill so that staffers like you can get to know real-life stories of what does it take for a startup to succeed, and what are the hurdles that hopefully Congress and the administration can help remove so that startups can grow. As I said, we started 25 years ago in San Diego, really because the leaders at the University of California, San Diego, and the leaders of the city of San Diego knew there was a lot of research and development going on, but they weren't seeing the kind of job growth and company growth that they would expect. And so they created Connect to help startup companies take that next step beyond the idea phase to the actual implementation stage and productization and commercialization. So we, over the last 25 years, have helped over 2,000 companies get started that are still producing and helping the economy. Sometimes those companies were merged or bought out by other larger companies, but they are still an impact on the community. And so just recently we opened our DC office and we focus on various issues related to innovation, startup company growth, small business growth. So going back to where San Diego started, we're fortunate that we have over 70 major research institutes in San Diego, both public and private. The Von Liebig Center is a newer kind of center that focuses on helping companies get started, and Dr. Ochoa is here today and she'll give you a story about what they do. We have a big defense and security industry in San Diego. The naval presence there is one of the biggest research labs in the United States for the military. Our wireless industry is growing. Qualcomm is one of the leaders there. Our life sciences companies, we have a growing biotech and medical device and other life sciences. The company we'll be talking about, Inflamagen, is one of those startups. Energy and environment. Our latest cluster that's developing of startup companies is in the sports industry. Obviously San Diego has a lot of people that like to get out and do fun things, and people are realizing, I can make a better surfboard, a better helmet, a better shoe, and I can actually build a company off of it. So it's really fun to meet these entrepreneurs who are your average folks you see out working out, running, biking, cycling, and then they're actually starting companies and having them grow. The other thing that we really have helped in the San Diego region is as each of these different industries start growing, they can sustain themselves in their own trade associations. So we've helped start Clean Tech San Diego, communications, sports, and last year we were fortunate enough to win the Economic Development Administration's Innovation Award for innovation in regional innovation clusters for our region. And as right now the focus in Washington, D.C. is so much on jobs and job creation, we really want to make sure that Congress and the administration is remembering that although we want new companies to form, we think it's more important to focus on tech sector companies. Obviously we need gas stations, we need cleaners, we need restaurants. But if you want high wages and you want well-paying jobs, the tech sector is where you can go to create those jobs. We found in San Diego that the tech sector is only 5% of the employers, only 10% of the jobs, but 25% of all wages in San Diego County. But those wages are 95% higher than the average industry wage. Those jobs also have a multiplier effect. where depending on which data you use, you can have anywhere from 6 jobs that go with every 1 tech sector. So again, it's not just the dry cleaners that opens, it's the scientist, it's the graduate student, but then all the other development that comes with it. And so we really think we want to help Congress and the administration learn about innovation and learn about these new companies. So without any further ado, I'll bring up Dr. Choa, who will talk about the Von Liebig Center and what they do.
Thank you, Tim. Good afternoon. The mission of the Von Liebig Center is to accelerate the transfer of university discoveries out of the private sector to the private sector and to prepare our science and engineering students for the entrepreneurial workforce. Through these 2 programs, we are aiming to increase the collaboration and the transfer of innovation between industry and the university. We started 10 years ago, founded with the support of the Von Liebig Foundation of $10 million. Since 2007, our center is supported by a combination of grants from the federal government, private foundations, and support from the university. We provide a combination of business mentoring by vision entrepreneurs, pre-venture grants for prototype, proof of concept, and graduate level entrepreneurial education. More than 1,000 students in science and engineering have gone through our program, our classes. We also are a forum that brings together innovators, entrepreneurs, mentors, students, and service providers. And we are a platform that can serve multiple institutions in a geographical region. And I'll talk to you a little bit about that. So where do we fit in the commercialization continuum? We actually operate at the transition between fundamental research out of the laboratory in the university and the early stages of commercial development. Today you will be introduced to Professor Gershon Schomburg and Mr. John Rollins. They started a company that was supported by the Von Liebig Center called Inflammagen. We spent $150,000 in grant funding to support the early stage development of the technology that led to Inflammagen. And also we provided business mentoring by an individual named Steve Flynn. Why proof of concept is important? Well, in order for you to launch a company that eventually will become a high growth company and create jobs, you need to move the technology out of the university laboratory. And it's not easy. Why? Well, because the technology that you find in the research lab is not ready for the marketplace. There's no demonstration that the technology works outside the lab. There's not necessarily a well-understood market for it. There's no business model, and there's no management. You have a professor with their graduate students interested in translation of that technology to the private sector. There's a culture gap between the research and the business. There's a different sense of urgency, motivation, and priorities. So our center provides an integrated approach to commercialization to help translate those technologies out of the university. One, we offer what we call pre-venture grants in the order of $50,000 to $100,000. These grants pay for the graduate students to do prototype, proof of concept studies in their laboratory with the assistance of the guidance of their faculty advisor and their business advisor. This funding is released on a milestone basis and is highly leveraged because it utilizes the university research infrastructure. We have 7 business and technology advisors. They are domain experts, seasoned entrepreneurs that understand also academia. They guide the researcher through the commercialization process, and they bring their connections and their network into the university to connect them to the researchers to the innovation ecosystem. We have 4 graduate level courses on entrepreneurship. And our aim is not to teach students how to become entrepreneurs, but really to think entrepreneurially and understand better how technology companies innovate and compete globally. Having said that, 10 of our students have started companies of their own. And they're very good at building business plans too, competitions. but that's not our objective. We have a well-characterized process for filtering and sorting projects and teams. So our resources and efforts are focused on those technologies that have the highest impact. This is the process that we follow. It takes about a year and a half to go through the whole process from beginning to end. From the moment that the researcher and their students apply for commercialization assistant, we assign them a mentor that will work with them throughout the year and a half, the process, preparing them, coaching them, and also giving them assistance to access to capital. We also assign MBA students to help the team, we call the innovator team, with market analysis and business plan development. When those technologies that are ready to leave the university, if they are integrated into a startup company, then now John can actually apply for an SBIR, CTR funding, or engage with angel investors, venture capitalists, and also they in some cases join an incubator. In 2007, we started with a number of initiatives in partnership with actually the city of San Diego, starting expanding the model to include other universities in the region. We have run 5 competitive programs that involve more than 150 researchers from 13 universities in Southern California. And what we do is programs similar to what I showed you in the process, but now focus on areas of interest to the region, like clean technology, wireless health, and innovation that reduce the cost of healthcare. So in the 10 years that we've been in operation, we have spent $4.6 million in grants for 82 projects. This funding has gone to support 100 graduate students and postdoctoral researchers to work on commercialization. 32 startup companies have formed, more than 200 jobs, and about 1,000 students have gone through our courses. So what have we learned through these 10 years of operation? Well, obvious for all of us is that still there is a huge gap between an idea and a product, and bridging the gap is not done by the researcher. We heard a quote recently that says that innovation without entrepreneurs goes nowhere. We agree. What we do is that we bring together the scientists and the entrepreneurs to join, to actually have a common purpose of towards commercialization. Researchers need business-oriented guidance throughout the technology validation process. A well-structured process that selects the best candidates and ties grant payments to achievement is critical, because that keeps the researchers and the innovators focused for 1 year towards achievement towards commercialization. The proof of concept process should focus on the role of the graduate student and the postdoctoral researcher. Many times we don't think about the professor, but it's really the students who do under the guidance of the faculty, who do the work, and also when they leave the university, join the company, either as CEOs or as employees. We also should expand the role that the proof of concept centers serve to support and leverage the intellectual capital that these very bright students offer in helping small and medium-sized companies with their solving problems or translating innovations from academia so they can remain competitive. Thank you very much. And
thank you. So my responsibility will be to explain to you some of the science. And what you will hear is probably new to all of you, the majority of you. I should say, we gave it a word called autodigestion. That word has been used in the past. I'm reusing it again, and you see in a minute why I'm doing that. The most important thing is this is only possible because we have an entire team, and that is supported through essentially 3 funding sources for us. All our basic science for many, many years, and otherwise I wouldn't be even standing here, was possible only by the involvement of the NIH. And then we wanted to translate this, and the Liebig Center, as Rosabelle just explained to you, played an important role, but then there was one very important next step. maybe the involvement of a venture fund that John was going to talk about it. Otherwise, this would not have been possible, what I'm going to show you. Now, the problem I'm talking about from a medical point of view is one of the worst in medicine. Namely, it's when you're in the intensive care unit, all kinds of tubes are into you, and a physiological process called sepsis or physiological shock plays out, in which it's been known for a number of decades now that by the time individual organs fail and you have, say, the third organ that fails, your life is now on the line. And the chances that you will leave that intensive care unit is very low. There are very detailed statistics to it. It is, in terms of mortality, one of the greatest problems to the United States and to mankind in general. And there have been many, many ideas that have been developed about this, and they all failed, with one exception. There's one treatment that improves the survival by 6%. And there's obviously lots of room for improvement. Now, I had the great fortune and the privilege with NIH funding to think about this problem for many, many years. And we have come up with a new concept behind it. And the concept is derived by thinking about your very unlikely candidate on the surface. But once you think deeper, you say, oh, yeah, of course, it has to be that way, namely your intestine. And so I'm going to show you a few science slides. When you eat or you just had lunch, it will accumulate in your stomach, and then it goes into this long ride through your intestine in order to eventually be discharged to the large intestine. That's the way we function, from day one to the last day of our life. At the junction between the stomach and the small intestine, there is one little portion called the duodenum, in which a key event takes place. Namely, in this little yellow region here, it's called the pancreas. There will be something mixed in. Your body mixes something in, and that's your digestive enzymes. And these digestive enzymes have one single task. All the steaks and all the goodies that you just had for lunch are supposed to be broken down into their what we call monomeric form. It has to be broken down so that the intestine can actually do it. So your intestine has the ability to break down just about all biological molecules. And you're completely justified to ask the question, this happens to be Chinese Sichuan style, if you eat an intestine, you can digest that intestine very nicely. You'll do it all the time. German sausage is the same, I promise you. And you should ask the question, is how come I don't self-digest? It's an interesting dinner conversation. This problem has been solved by nature long before us. The dinosaurs had already had one, so it's a very old and very robust process. And what many, many years and many publications have shown us, the way nature has solved this problem is in the following way. You're looking at a cross-section of an intestine. Here in the middle, where I wrote down the name of these enzymes, which are your digestive enzymes coming from the pancreas, this is where you digest. This dark layer of cells and structures that you see here is where you absorb. So a normal, healthy intestine will keep all the digestion compartmentalized in the lumen of this intestine. So the whole ride along the length of the intestine. You're not supposed to ever have any of these digestive enzymes inside the wall of the intestine. They're not supposed to be here. And usually, they're never there. The reason that's possible is because there is a layer of cells that is shown a little bit more magnified here. Each of these is called the villus structure, and each of these is now lined by these cells here. Each is a cell that tightly packed together to make sure that none of your digestive enzymes actually make it into the wall of your intestine. These cells are very similar to what you have on the inside of your cheek. So you're very nicely protected there. And nature has made sure, through very clever mechanisms that we understand to a great degree today, that these digestive enzymes that are outside in the lumen, up here, don't get down here. Now, anybody who ever studies membranes in biology, there's thousands of people across the country, they would tell you, yeah, this is textbook knowledge. This works usually, yes, and textbooks will probably tell you that, but that's not the reality. And indeed, when we started to look at this barrier, as we call it, in the context of shock, we discovered that, in fact, the world is not so simple. There's something much more, and actually more catastrophic, that occurs. You're looking at a slide that we prepared, which shows you by the green color the intensity of that enzymatic activity. And in a normal intestine, this is this villus structure that you saw before, there's little of that, usually between the villi. But if we disturb that intestine, and there are many ways to do that, what you start to see is that these digestive enzymes get into the entire wall of the intestine. It's all green. And what these enzymes are doing is they do what they're designed for. They digest. I don't care whether this is a steak that you just ate or whether it's your own intestine. They just continue. You have them in very high concentrations in your intestine. And what they do is they start to digest your intestine. And the results are catastrophic. First of all, this nice little structure here gets completely digested away. So the barrier is gone. And you're now completely exposed to your own digestive enzymes. Nothing holds them back anymore. And the next item is that you will produce additional products. So this is sort of a summary sketch of literally hundreds of studies in which this is normal situation. All the nice little Chapman here, the digestive enzymes are nicely compartmentalized. They stay in the lumen and in the wall of the intestine. Only the stuff that got absorbed through the epithelial cells gets in there. But by the time the wall is disturbed, these Chapman get into the wall, and now they take the wall apart, and they produce secondary products. Themselves are toxic. And the result is, if this stuff gets carried away into your bloodstream, which it does, you will now go in what's called multi-organ failure. In other words, the digestive enzymes, together with the fragments that are being formed, get into the rest of your circulation, and the consequence, your lung will start to fail, your kidney will start to fail, and it's only a question of to what degree this whole thing plays out. That story that I just described to you in a nutshell is essentially probably the way most of us will die. And the question is there in an acute situation a possibility we can do something about that? And the obvious issue is well, we have access to this particular compartment because all we need to do is put down a catheter through the esophagus into the stomach and right at the entry of the small intestine, and we can place through that catheter any inhibitor that we want to. And that's indeed the idea that we developed as an intervention. And we allow these different types of enzyme inhibitors, many of them are actually existing drugs, to go down over the whole length of the intestine and make sure that we block that digestive tract as much as we can possibly do so. Okay? So this is an entirely new approach to shock and multi-organ failure. My laboratory is one of the few ones in the world that has published extensively on it. There's only one other laboratory in the world that has now picked up with this and published a similar paper on the same issues and have confirmed it. So you are on the cutting edge of this development. This is not public knowledge yet. And so we want to show you just one result. This is how a normal intestine looked like. I defocused it a little bit, so I hope it's not too offensive to most of you. This nice, smooth, blue-looking organ. This is how an intestine looks like that has been, comes from an animal that was in shock. It's bloody, it is disrupted, certain parts of it are swollen, and it's a non-functional organ. That is the way your intestine will look like on your deathbed. This is how the intestine looks like when you place these inhibitors in the lumen, which is right here where I'm pointing at, of the intestine. And you see it's not completely preserved, but it's dramatically better than what this looks like. That intervention we are testing now in a number of ways. And one of the important things we had to have in place, and this is where funding from a private venture was necessary because it's too early for clinical trials. You wouldn't get any funding for that. Is to do long-term survival studies. Does this really make somebody survive longer? And this is one example of a whole long sequence of studies that we hope to publish shortly, in which we show that if we block with a known drug, an existing inhibitor, the proteases, we can go from a survival of only 25% to a survival of 83%. So there is a dramatic improvement in the ability of these animals to survive. And these are long-term survival studies. And we have many such similar results. So what we had at this particular point, what I had insight to, by the way, we'll be able to do this on another species other than the rat. We also got some results on a pig. And so I had the very first frontier results in my hand. I have the ability, I was very confident, to save lives. Countless lives. And the question is how do I now translate this? Okay? Lots of people very skeptical because shock research had nothing but failures, and therefore many people said, no, we don't believe you. And the only way I can convince people is if I show some human results. And so as a result, I was able, through a fortunate coincidence, to get in contact with China's premier heart transplant surgeon. His name is Jing Wei. He works at a private hospital in Taipei. And Jing came to me and said, I have these very high-risk patients. Could you not come to, he learned about this procedure, read all the papers, and said, why don't you come to Taipei? I did that. I spent a day with him, giving nothing else but the seminar, where I explained in great detail many of the steps I just showed you from a bird's eye point of view. The afternoon, we talked in detail about how to deliver those protease inhibitors. We went for lunch, for dinner. At dinner time, one of his surgeons called up and says, I have a septic patient. And this was an individual who had a transplant in 2006, was immune suppressed, had a car accident in 2010, and was self-treating, and then finally failed, had a terrible destruction of his large intestine. It was just end of line. And the physician asked me one more time and says, okay, Gerd, tell me, what was the exact concentration of the digestive enzymes that you gave? We recomputed again. They had the drug in Taipei. It was approved for a completely different indication. And he administered that drug that night. That was a night I didn't sleep. The first patient in the world to be treated. Now, this poor fellow had indicators of shock, which are about 10 times higher than what they're supposed to be. And so the physician gave it on a consent basis and just said to the family, you know, for your loved ones, unfortunately, we have nothing else to offer anymore. But we just learned from a purely experimental procedure has never been carried out in man that we would like to offer you to do that. And of course, the family gave a consent for that because the alternative would have been death that night, probably. And much to our surprise, this patient, within a day, was able to bring down these amylase values. They maintained low while they blocking this digestive enzyme. So this is the first patient in which all the digestive enzymes, I shouldn't say all, most of the digestive enzymes were inhibited as good as we could and knew at the time. He had to go through a long, lengthy healing period. Eventually, his white cell count came down to what is normally present in humans. This was about a month later. If you look at his intestine, I don't know whether you can see this. This was his intestine on that day. It was in terrible shape. It was completely distended. There was lack of proper motion and absorption and so on. So it was an intestine of typically a person you would find in the morgue. And this is how his intestine looked like about 2.5 weeks later. And he was released from the hospital at the beginning of November. And the experts, I mean trauma surgeons who looked at this case are all in agreement that he shouldn't be alive because he had a severe sepsis, was immune suppressed, and also on top of that had some very bad hemodynamic results. This case was just presented as a single study case at a transplantation meeting in Korea. It is, and it will be published in the proceedings of that meeting, hopefully within the next couple of weeks. So this was a starting point. Professor Wei, in the meantime, has treated a total of 13 other such patients where he had these very high indicators of sepsis. And out of these 13, now this is not a statistical trial. This is just individual consent cases. He was able to save the life of 11, and 2 of them passed away. And in fact, I know pretty well why those 2 others also passed away. They had a problem that we did not address by just enteral treatment with this type of approach. So it just illustrates to you that it's, for me, a challenge to translate these ideas without spending decades convincing my colleagues from preclinical studies that this is a worthwhile adventure. And therefore I have difficulty, I'm able to get funding, and I want to highlight this, from NIH to do basic studies. Without the NIH, as I said, I wouldn't be standing. But when it comes to that next important step, I don't know how to do it. And I find my clinician colleagues, whom I have many, they say, this is too risky, not ready yet. You need to do some more experiments. And the funding situation is of such a nature that the funding agency says, hey, listen, you've got to do first some additional studies before you are ready, and you need to answer the following questions first. So we get a long list of questions. In the meantime, I know I could save some lives. So this is where it's a real valley of death, and not to talk about just the funding itself. John will give you some more details about that. All of that funding is necessary to pay these people. There's a large number of people who have participated in there. Included, I want to mention briefly, because you may know his name, David Hoyt, who is currently the executive director of the United States College of Surgeons in Chicago. So I want to thank you for your attention. And the message from my point of view is that I'm a basic researcher. I need to keep all my eyes focused on the basic mechanism. I can't spend my time otherwise. I'm 100%, 150% occupied with just making sure that the science is right. I need other help from the university, including the Liebig Center and fellows like John, to actually try to translate and bring this to a benefit for humans. And I feel, because I published the first paper in the year 2000 in the Proceedings of the National Academy of Science, it's not just your backwater type of publication, that the translation has taken way too long. I know, and I'm putting my reputation on the line, I could have saved a lot of US soldiers' lives and congressmen's lives. But it didn't move. So, John.
Thank you, Gert. Well, first of all, let me tell you who I am. I run actually it's John Rodenreich, it's not like it's spelled. I run a venture capital firm. So I know you're all rolling your eyes and saying, does the world need another VC firm? Because we've got a lot of them and they're just parasites on society. But we're a little bit different. Most VC firms invest in what's called late stage companies, where they've got revenue and there's low risk. What we look for are disruptive technologies like Gert's that we can license from a university or a federal research lab and then commercialize. So we're willing to take high risk for a higher reward. Now, Gert gave you a very detailed description of what sepsis is and what the science is behind it. Let me tell you the magnitude of this issue. We're going to have you captive in here for about an hour. So in that hour in the US, 24 patients are going to die from this. So it's not like this is a minor flu incident. This is a major unmet medical need. And Gert mentioned that there is one drug that increases survival by 6%. That drug costs $6,400 a dose. So every time it's given, CMS reimbursed at $3,400 bucks. So it's a very expensive drug for little benefit. And if you remember Gert's animal statistics, he's got about a 60% improvement in survival. So that's about an order of magnitude increase. So About 1 million deaths a year worldwide, a little over 200,000 in the US. In 2007, which is the latest date that we have data, US hospitals spent about $38 billion just on sepsis. And pharma companies have spent billions of dollars on this. They've run over 100 studies, and there's no real solution to this. And there wasn't until Geert came from left field with a different way of thinking. So it's a major problem. Now, Roosevelt talked about the valley of death. And this is the difficulty that a technology like Geert's encounters in trying to get something commercialized. And one of the major impacts is it's hard raising capital, both from the government and from private sources. Now, Geert's been funded a lot by NIH and NSF. Well, there's talk now of cutting their budgets because of the whole budget crisis. So the capital raise situation may get worse. But you can throw all the capital you want at these kind of problems. And in life sciences, that's not the whole issue. You've also got an issue of how long it takes. Now, what I'm going to do is contrast what's happened in the US versus what's happened outside the US in terms of trying to move this through. So in addition to money, the time it takes to get this through is also difficult. It's like a boa constrictor swallowing a rabbit. It just takes lots of time to get through there. So I put the FDA up there. Now, you all know the FDA takes forever to do things. Sometimes it does, but I'll show you an example where they've been reactive. It's not just the FDA. If we want to treat a patient in a US hospital, we have to go to what's called the Institutional Review Board. And their mission is to protect the patient. So they're not real interested in using his technique because it's not standard of care. Because here's the deal. In the US, if a surgeon used his treatment, and remember, his treatment is based on an approved drug. The drug's already approved. All he wants to do is put it in the stomach instead of IV or have somebody take a pill. That's what his treatment is. So if they use it and the patient either dies or gets worse, guess what happens? They get sued. That's not true in some of the other countries. So they're somewhat reluctant to say, let's get this approved. He's done extensive animal studies, 300 rats, a couple hundred pigs. To get the Animal Subjects Committee to approve a long-term survival protocol was excruciating. So it's not just the FDA. There's an attitude here in the US about, do we do innovation and take some risk or do we not? His treatment, which is for a disease that's 30 to 6% fatal in a hospital, is treated the same way as a treatment for acne. So there's no urgency here. It's, if there's any harm that can come to a patient, they do not want to approve it. You're all in the government. You know about bureaucracy. I know there's none in Washington, but in hospitals there are some. We have to deal with lots of different departments to get something done. And I'll give you an example of that. And lastly, it's difficult to take myself, who's a greedy capitalist, who talks about EBITDA and balance sheets, and connect with Geert, who talks about gel zymography and pancreatic enzymes. So when I first met Geert through the von Liebig Center, we spent a couple months trying to get to know each other and talking about, okay, what's this science? And I finally went to Geert and I said, it's like you're speaking French and I'm speaking Greek. So what do I have to do to communicate with you? He said, it's pretty simple. You have an engineering degree. Pick up the molecular biology textbook and read it. So I bought it from Amazon. I get this huge package. I said, what is this? It's 1,740 pages of dense molecular biology. I said, well, if I really want this, and I feel this is a breakthrough technology, I need to understand the science. So I literally spent 6 months reading that book. I don't know of another VC who would take the time and energy to do that. If I hadn't, we would not have made a success out of this, because I would not have understood it. So there's a difficulty in taking somebody like myself, who's from the business world, in getting them tied up with somebody like Geert. We talked about timelines. On the device side, timelines have increased significantly since 2007. 45 to 75%, depending on whether there's an existing device out there or whether it's a brand new one. We're working on a therapeutic compound, a drug. We're using an existing drug that's approved. So we can follow what's called this 505(b)(2) timeline. All I'm going to talk about today is what it takes to get it from animal studies into where we treat humans. So that's the only part of the timeline we're going to talk about. Now, one of the issues I faced, here I was, a business person. He works for University of California, San Diego. Takes in almost $1 billion a year worth of research funds. One of the premier research institutions in the US. But it's got lots of departments. Garrett is in the Jacobs School of Engineering. His colleague, David Hoyt, is in the School of Medicine. Well, UCSD, and I don't want to insult UCSD, but there are two fiefdoms here. They don't always talk to each other. It's pretty typical. Worse in this case, we had a biochemist who was at a third institution, the Torrey Pines Institute. And to make it worse, if I wanted to talk financial terms, I couldn't talk to Garrett. I had to talk to technology transfer. So in terms of me coming from the outside, I run a fund, I've got a board and investors. What we wanted was we wanted the IP, we wanted the patents, so I could commercialize it. The university wanted the money. I literally didn't know what to do. What department should I go to? How should I approach them? So that's where von Liebig came in. The von Liebig Institute, Roosevelt introduced me to Steve Flame, who's one of their advisors. And he helped me understand the science, and he helped me navigate this morass of different bureaucratic departments. Without von Liebig, this wouldn't have happened, because I would have given up, or I would have failed at it. So what we did is von Liebig gave us some money. We got to the point where we knew it worked in animals short term. And I said, Garrett, I'm not going to spend a lot of money on this until you can show me that it works long term in animals. So we gave Garrett some funding, and we did long-term rat studies, and that we had some concurrent pig studies at 2 different institutions. So we finally got to the point that Garrett described. This thing works. Works in rats, works in pigs. Let's go and use it on humans. Trouble is, that takes a lot of money and a lot of approvals. So this was 2008, and here's where we faced the perfect storm. We needed a lot more money, and we needed approval to do this. It's an existing approved drug. Rather than use a tablet, I want to mix this into a liquid and put it in somebody's stomach. That's all we wanted to do. So we went to the NIH. They said, no, this is translational research. We fund basic research. I said, all right, well, I'll go to my friends in the VC community, and I'll go to angels, who also fund early stage. Remember, this was fall of 2008. You remember Lehman Brothers and all that. They said, well, not only is it too early, we don't have any money anymore. So we lost it all in the stock market. So I said, okay, I'll go to the pharma companies. They got lots of money. They need new therapies. They said, no, you got to get this in humans first. So it's like Franz Kafka in the trial. You're caught in this circular situation. So I don't know if Garrett mentioned this, but he had gone to the Navy in 2000. They weren't interested. But we had met some Navy people. So we went to the Naval Medical Center in San Diego. It's a hospital that treats military personnel. And we met with them in 2008. And they said this idea has a lot of merit because it could help soldiers on the battlefield. So about 9 months later, they submitted a grant. The grant finally got approved. And testing started earlier this year. So this is for pigs. We're talking around 3 years. So then we talked to the Veterans Administration Hospital in San Diego. Garrett has a colleague there who knows what he's doing and said, we think it's time to start treating in humans. So early 2010, we wrote a protocol. We started developing it. It took us like 9 months because when you're dealing with humans, you got to dot your I's and cross your T's. So it took us some time. We then submitted it to the IRB. IRB looked at it and said, Garrett, you're probably right. I'm sure you could save a lot of lives. It's an existing drug. I'm sure it's safe. But it's too risky for us. What happens if something goes wrong? So they said, you've got to get FDA approval for this. So at that point, I almost threw up my hands because I said, okay, I'm going to face a year trying to get through the FDA. So we wrote what's called an IND exemption. We went to the FDA and said, look, we shouldn't have to go through a full new drug approval. It's an existing drug. Give us an exemption. So we submitted that in late May. And I said, okay, I'm going to take a vacation for a year because we won't hear from them. Well, 6 days later, they approved it. I've never heard of that quick an approval from the FDA, but it happened. So they're not always the bad guy. This was a case where they looked at it and said, this is something that's needed. So 2 months later, the IRB said, okay, if the FDA is okay with it, we'll approve this. So go ahead and start admitting patients. Get the pharmacy ready. So we started doing that. And all of a sudden, in the bowels of the VA, there's a separate research committee. And they looked at it and said, I know it's already approved, but I don't like the wording on the HIPAA form. So they said, you're going to have to go back to the IRB again. This just happened like 3 weeks ago. So right now, we don't know what's going to happen in terms of when we're going to get into humans. Now contrast that with Type A. July 31st, he trained him. They treated the next day. The difference is they didn't worry about tort issues or getting sued. Their focus was on the patient. So do that timeline comparison. 3 years for pigs, 21, 22 months in the US, and a day in Type A. Let's look at the difference. Here are some of the gating factors we ran into. It turns out that our relationship happened pretty quickly, probably because we both had the same colored hair. The tech transfer office was quick. Animal committee was slow. We had difficulty getting any kind of money. But with the FDA, we were very, very fast. So if we'd adopted Type A's timeline, I calculated this out. It's 475 days, said to today, from when they first treated a patient. If you look at what has happened in the US, that's 279,000 deaths. We couldn't have saved them all. Well, what if we could have saved 10% of them, or 20%? It's $50 billion in hospital bills. We're certain we could have saved a fair amount of those. So how can you folks help? Well, first, this would not have happened without the von Liebig Center. So we think that if you're a major research institution, you need to have a point-of-care center. And HR 1036 incentivizes the funds that U.S. corporations hold overseas to invest in innovation. I mean, U.S. corps have about $1.5 trillion sitting overseas. They're not repatriating because they don't want to pay the 35%. Well, this bill says, let's give them some incentives to bring it back, as long as they invest it in something that's going to create jobs. The FDA looks at us the same way they look as a treatment for flu or for acne. There should be a different mechanism for a life-threatening disease where it gets approved quicker, or at least looked at quicker. There's several ways we can increase early-stage revenue. It's difficult in the US to get money for these kind of early stage. So maybe there are things we could do with taxes. Maybe we can increase the Reg A limit so that small IPOs are easier. And there's a bill, actually 2 bills, one in the House, one in the Senate, that will do that. And you can have copies of this if you want. You don't have to copy all the data down. Tort reform is important here. The reason the US acts differently is because physicians are afraid of getting sued. We did not use the SBIR because we're a VC firm, but many of my colleagues do. It's a phenomenal program. The trouble is, we don't know what's going to happen to it. There's a continual resolution every 3 months or every month. So there's a lot of uncertainty. So we think it should be reauthorized permanently, and we also think that it should allow VC-backed firms. So if we do this, first we're going to create a lot of jobs. If you look at the last 18 years, 42% of the new net jobs were created by companies of less than 100 people. If we can spur these kind of innovation in these innovative companies, we're going to create a lot of jobs. We're also going to reduce healthcare costs. And with Obamacare, that's going to become more and more significant. And finally, we're going to save lives. So thanks for listening. I hope we can work together and reduce this valley of death and save some patients. So I've turned over to Tim.
So one of the things we wanted to make sure that people realized was that the market forces are hard enough for a startup company to get going. You've got to distribute, develop, manufacture. All those things already exist as barriers to startup growth. And what the story we hear today are that there's even more barriers, whether they be through the university system, through the governmental regulatory system, the legal system. And so our main message is just to try to educate and help encourage the focus to be what are laws we can change, what are regulations we can change to remove some of these barriers so that these companies have a quicker access to the market and have a better chance to create jobs. We have a few minutes left if anyone has any questions. Any of us are willing to answer them, and we can also stay afterwards and chat as well if anyone has any questions. All right, well, I'm going to take a moderator's. Do you have one?
I'm just curious about the funding levels you were talking about here in the Valley of Death. What type of numbers are you looking at?
Valley of Death, what numbers? I think it depends on where you are.
In terms of what we put into this,
Yeah.
we put a total of about $3 million in, which is a drop in the bucket when you're talking about taking this kind of treatment and moving forward.
But are you asking where do you see the Valley of Death?
Yeah, I mean, that's your initial money in terms of what you're looking at from the Navy and VA. Is that included? Or is that
No, that's different. The Navy got a grant for $600,000 to do the pig testing. Garrett got probably $6 or $7 million with NIH grants initially to move it forward to there. When Von Liebig gave them $150,000 or so, when we first licensed it, we didn't pay very much to the university. It was like $25,000 or $30,000. And we've been supporting his lab at about $200,000 a year. Very small amounts of money to pull something that's really important through this valley of death.
And how much do you think you need still going forward? Because you're still not through it yet, right?
We need about $4 million to move it into phase 2. We've got one phase 2 that's going to go on to the VA. We're going to do 2 more. That gets us through what's called efficacy study. Does it work? To get this drug approved, we still have to go back and do what's called a phase 3. That's a couple thousand patients. It's multi-site. It's probably $25 million. We probably can't do that. We're probably going to have to partner with a large pharmaceutical company. But it's probably $25 million and 18 months or so to get that through to where we can file a new drug application. So we're probably looking at 2 years before we can treat people in the U.S., which is frustrating.
Heard for some pharmaceutical startups, and the time they think of the drug to when it finally reaches the patients, it's something like 15 years or something to that effect, maybe 20 years. But my question is, how does that compare with other countries? I don't know, Japan or Europe. Is it significant? Are we talking half that time? Or is it roughly the same or longer?
It's not roughly the same. It's longer to take a new drug than it is to take an existing drug, which is what we've done.
Okay.
You're talking probably 8 to 12 years in the U.S., if you're lucky, if you don't fail along the way. If you go to somewhere like Eastern Europe, you can probably get it done in 6 or 7. But they follow the same kind of pathway. In fact, to get this treatment approved in Taipei, we're going to have to go to their equivalent of the FDA and follow the same kind of process.
Can you elaborate on your experience with the military? You talked about getting this drug funded to some extent by the VA and the Navy. And then, did I hear you say you're in trials, phase 1, or what's your next step with?
Well, with the VA, they have put a proposal in to do a full phase 2, which is we're going to take septic patients come into the ICU, we're going to randomize them. Half of them are not going to, they'll get a placebo treatment. Half will get the treatment. It's a 200-patient trial. We're going to pay for that. And hope as soon as we can start, we'll start it. We've got the funds. We want to do 2 more facilities. And we have more hospitals lined up for this than we can really accommodate. We're looking at about $4 million to do that, and maybe a year and a half. Beyond that, it's the $25 million I talked about for a phase 3.
So the studies done in Taipei were consent studies as opposed to trials and stuff like that. When you bring that to the system over here in the States, and the time it takes for clinical trials and stuff like that, is it similar to comparing apples to oranges where you could do consent studies here more rapidly, or is it kind of the same?
Precisely the point. See what he's saying, a physician in the U.S. can take a legally approved drug and use it for a different use if they think it'll benefit the patient. That's what they did in Taipei. They won't, because if something goes wrong, they'll get sued. That's the difference.
Strictly adhere to FDA rules, I think that's a national patent for them to make sure that there is no violation or no unethical behavior. That's highly ethical. I know where the breaking I know where that is. So they're not trying to show something, but they're not afraid of a lawsuit in a situation like this, when there's very few other chances for the patient. There's a big difference.
Explain what role like intellectual property has played in this also, whether there's a patent involved and the importance of that.
We invest in the early stage technologies. We only invest if there's a patent, either filed or in place. In this particular case, UCSD tried to get a licensee. They had an issued patent from 2003. Nobody would touch it. I found out about it after listening to their talk. So we licensed the technology and the patents. And we've subsequently filed 14 different patents around this in terms of dosing, how we do it. So IP is critical here. Without IP, we would not have invested. And don't get me started on the U.S. Patent and Trade Office in terms of times. That's a different discussion.
Well, thank you all for coming. There's material at the back. I'm here full-time in Washington, D.C. We opened our office a year and a half ago. I'm happy to come back to the Hill and meet with any of you or get you connected back to them or other companies the Von Liebig Center is working with. Thank you all for coming.
Good presentation, Jim.